so we're going to get started. So this is the Longevity 0 to 1 course. Hopefully everybody is in the right room. My name's Nathan. I'm co-leading this course with our wonderful co-host and organizer, Cat. Yeah, maybe Cat, you can introduce yourself.
Hello, my name is Cat, and I'm part of VitaDAO. We also work on this course with our colleague Mark, who unfortunately cannot be here. So this course will take you from zero biology to completely curing aging. So by the end, we will solve the problem. We can all go home. What do we think is the probability that one person in this room will live until 200 years old? Anyone has a guess?
Shout out and answer. Yeah.
Above 90%.
Above 90. Okay.
Wow.
Okay.
Optimistic.
Any other takers?
Are we talking now or in 10 years' time?
A person in this room will live until
Right.
at least 200.
Can also be here in 10 years' time.
Oh, thank you.
Anyway, who's the youngest person here? That affects the data, darn it. Is there anyone below 10?
It depends on what we do.
Okay.
It depends on what we do. It's a very good answer.
Yeah. Good.
Do nothing, 50-50.
Okay. Do something.
Do something, close, like asymptote to 100.
Okay. Awesome. Okay, great.
Love the
Who in
optimism.
this room doesn't drink alcohol or eat processed sugar? Okay, so now it's like people. There you go. It's 25%.
25%.
Yeah.
Wow. Yeah. Actually, when we ask these questions, even like in the field, people have really wide answer. The highest that I've heard is 80%, and the lowest is less than 1%. So, the answer is, I guess we just don't know. But I think the agreement is that it's possible to cure aging, but the fact is that nothing so far has been known to increase the max human lifespan. So coming back to Lauren's point, if we don't do something, if we don't start doing something, the probability is very low, and we have to start acting right now to do something. And Nathan, do you want to talk about this?
Yeah. So people are working on this problem. So that's the good news. And hopefully some of you in the room will join this industry and try and tackle this problem of aging. So there's been a lot of interest in this space and a lot of headlines that you might have heard of. So in the last year, about $4 billion went into like private investments in companies trying to develop, cures for aging or therapies for aging first in the longevity biotech industry. One of which, of course, some of you have heard, like Altos Labs, right? Made a lot of noise last year, but it's still a really small industry compared to the size and scale of the problem that we're trying to tackle, right? So aging affects everyone. Predominantly, of the top 10 causes of death in the United States, about 70% of them have a very strong age-related component. They're either age-related diseases or correlated to aging. But despite aging being such a big problem, causing a lot of suffering and death, very few people work on it. So you can estimate that the entire aging biotech industry and academia focused on fundamental biology of aging is probably 10,000 people or so, in that sort of order of magnitude. So we need more people.
And that leads us to this course. So we don't want anyone to be a hobbyist in the field, and I think aging has gained a lot of popularity in the recent years. But most people who join what we call longevity hobbyists are people who want to see what the cutting edge research is, what they can apply to their own life. But we need people to be a lot more involved, and so we're creating this course. We want to give you an overview of the science, engineering, and all the trends of different interventions, and also point out what are the high impact areas that you can use your existing skills to work on. What we are not trying to do is to teach you to be a biologist, because that's that takes 10 years, not really something that you can do quickly. And also, frankly, longevity needs a lot of other skills in it, not just biology. And we also are not covering diet, lifestyle, and supplements. Those are very, very good things to follow in your personal life, but like to really radically extend your lifespan to beyond 120, I think we need more radical interventions. And this is what the syllabus looks like. There are 12 sections. For this course, the order is a little bit different due to speaker schedule, but this is a general idea. Right? So we have 3 classes, that crash course on the science foundation that you will need to understand and think about all the other parts that come after. And then we have 3 sessions on the practical ideas of running a longevity, genomics, or biotech company. And then we have 3 sessions on different strategies to cure aging and areas that you could work on. And then the very last part is on actionable opportunities. I think a lot of people in this room comes from an engineering background, so we have a strong focus on bioengineering and on computational biology. And then we can also cover alternative paths like cryonics, just in case.
Okay, so main takeaways. So people talk about existential risk. Aging is probably the one that we have the highest confidence in terms of potentially killing you or being sort of that last cause of death. So it's very deterministic in that sense. We don't know anybody who's been able to dodge aging. Okay, and then follow up to that. The good news is people are working on this problem. There's a whole field of biomedicine and longevity biotech trying to develop interventions to intervene at the fundamental mechanisms of aging so that we can live longer and healthier lives. And we know that there's nothing in principle that could prevent us from curing aging, although this could be an extremely difficult problem. It's probably one of the hardest problems to solve. But there's other problems in the longevity field that we'll cover in the bottleneck section. So we'll be able to give you a good overview of the fundamentals of the space, the science, the industry, and the bottlenecks. And hopefully some of you will be able to contribute to that after you have a general understanding of the context, and you can go further in more detail on your own once you have sort of a map of what's going on. And I think the last thing that we really want to stress is we don't want tourists in this space. What do we mean by that? We want people to contribute in some sort of way. And it doesn't have to be, I need to drop what I'm doing today and quit my job tomorrow and join the longevity biotech industry. It doesn't have to be that, but there's all sorts of smaller ways you can get involved, either through advocacy, spreading the word, and then slowly dipping your toes into this space and learning more, finding different ways you can contribute on a technical level or on an entrepreneurial level or an investing level. There's tons of opportunity because it's such a small space. Any one person in this room could have a really big outsized impact.
So I think we'll get started soon. So the schedule for today is this session is about general biology. You will learn all of biology in the next 80 minutes. This isn't a very easy task.
So my name is Boris. I am finishing up my PhD in molecular genetics when I'm not doing D-size stuff or stuff with Gitcoin or being at Zuzalu having a great time. Yes, I definitely can. There we go. So I got asked to talk about the sort of fundamentals of biology at the cell and molecular level, try to give you a crash course of some of these key topics, maybe set up the next few days. So I guess let's get started. Also, if anyone has questions at some point, put your hand up and just ask, because this is really, really basic stuff. Obviously, there's not too much I can cover in 20 minutes, but I'm going to try to make sure the key players and the key terms are explained. Hopefully by the end of this, you'll have an understanding of how it all fits together inside a cell, and maybe have some appreciation for the deep complexity here. So actually, what I'll start by saying is aging is a very complex process, of course, and if we want to be able to cure aging or fix aging, we need to understand what the non-aged or basic normal process is. So if you think about a disease is sort of biology being broken in some way. And of course, if you want to know how to fix that, it's really good to know how it works normally. So we can start off with cells. So all living things are made up of a single cell or multiple cells. cell is the basic unit of life. And it's kind of special because it separates the outside world from the inside world. As you can see on the outside here, we have a plasma membrane, which creates the outside of the cell. And then within the cell, there's even more membranes which create these subcompartments. And this spatial separation is a very important concept in biology because it lets things be regulated and separated. If everything was just like in one vat mixing together, like we'd just be like the primordial soup, we wouldn't have these amazing self-organizing autopoietic systems which really like create themselves from themselves. Like every cell came from a cell. So I'm not going to talk about the origins of life, but just talk about a sort of like stereotypical human or animal cell here. There's some stuff on here that will probably come up later on in the lesson, but I mean, we can start off with talking about the nucleus, which is where the genome is kept, and this is often thought as like the control center of the cell. And as you expand outwards, you see things like the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus. This is all machinery that helps cells create proteins and secrete them or export them to different membranes or out of the cell, and get them sent to where they need to go. Basically, we're going to spend most of this talk talking about different biomolecules, the different parts that are inside a cell. Because basically, a cell is a really, really, really complex machine. I mean, like I like to think of people as meat robots. We're just like super complicated meat robots made of trillions of cells. And the reason why I find biology so interesting is because everything we're talking about here, biology at the cell and molecular level, it is happening inside of you trillions of times per second constantly and allows you to understand and perceive all of this. It's like knowing what's happening truly at the molecular level that makes you what you are. Okay, so biomolecules. We can start off with proteins. Proteins are basically the machines that make life happen. They're like the tiny, tiny molecular machines at the sub-nanoscale that are constantly moving around and doing things. And when they're doing things, they can be providing structural support to the cell, but they can also be doing things like catalyzing chemical reactions as enzymes. Enzymes are just proteins which have a specific function. But basically, you can think of proteins as the parts that are actually making everything happen. The other important type of biomolecule would be nucleic acids. So DNA, all of your DNA is collectively known as your genome. You can talk about genomes sort of on a population scale, like population genomics, or you can talk about a single organism's genome, or even a single cell's genome, if you want to get really, really granular. And yeah, DNA is essentially like the code that makes all of this possible. It's, we'll talk about genotype to phenotype afterwards, but that's kind of basically it. It's like this is the instructions for the cell to do everything that it does. RNA is a related nucleic acid, and it is sort of the intermediary between DNA and protein. We're going to get to that in the next slide. But basically, you go from DNA to RNA to protein, sometimes from RNA back to DNA, but RNA is a very similar molecule, except instead of just being the code for your genome, it can also have actual functional roles, sort of like proteins, but not proteins. We also have lipids. So I talked about the plasma membrane and all the various membranes for organelles. So these are lipid bilayers, and I feel like everyone here has probably seen this at some point in school. I don't have a nice diagram of that actual molecule because we're not going to spend a lot of time on it, but this is what makes up the membranes that allow us to separate inside from outside. Finally, carbohydrates. So I'm not teaching the metabolism section, someone else is, but I should just say that carbohydrates are not just used for energy and metabolism, glucose, etc. They're actually also used in lots of different ways. For example, proteins can have carbohydrate molecules attached to them covalently, and this changes their function. Or maybe and that can mean a lot of different things, whether it's its activity or where it ends up going or how other proteins recognize it. I mean, carbohydrates can be on the outside of the cell as well, which affects how cells interact with each other. And finally, we have small molecules, ions, vitamins, metabolites, everything else. But I mean, if you think about a cell, it's like, I think 70% protein by dry weight. And then a whole bunch of other stuff constantly floating around. So there is this concept of the central dogma of molecular biology. Show of hands, who here has heard of this, like this specific wording? Okay, yeah. So there's a longer story about the central dogma of molecular biology when it was first coined and exactly what it is supposed to mean. But At a high level, it essentially describes information flow in biological systems. Because we were thinking of DNA as a sort of code and protein as the functional output of code in a simplified diagram. We can look at it like this. Essentially, we have DNA, which gets transcribed to create an RNA template, and then that RNA template is translated to make protein. There's a few other little loops here added in, a little more detail here. For example, there are enzymes called DNA polymerases, which will actually catalyze the replication of DNA. So I'm not going to teach you about evolution today or the semi-replicative nature of DNA, but this is essentially what enables evolution and allows cell division, because DNA has two complementary strands which can be split apart, copied, and then you have two copies of that DNA. We'll talk about DNA in a little more detail in a second. But I think it's also important to remember that we're really talking about physical systems here. We're talking about molecules. This isn't like code in a database. It's not just like bits in some sort of system. This is actually like physical molecules that life does computation on, essentially. Like there's instructions for making physical things encoded in this physical thing. And there's all this machinery, protein machinery, that enables all of those steps to happen. I maybe had this in my notes for a previous slide, but I mean, the thing that I want to maybe explain here and help you appreciate over this talk is just the complexity of this. I mean, your genome is 3.1-ish billion base pairs, and that genome, so a base pair is like two DNA nucleotides, which we'll get to in a sec, and every single one of your cells has that genome, which is kind of crazy. Like that means that there's about 2 meters, if you've strung this DNA molecule out, end to end, it's about 2 meters inside every single cell, and that's like all packaged together. And like there's trillions of cells in your body. We're talking about a nucleus, which is maybe like, I don't know, 10 micrometers wide, and we have all of the DNA condensed and compacted and packaged in there, and it's constantly being like copied with DNA polymerases, being opened up and copied to make RNA transcripts using RNA polymerases. And this is, again, very, very simplified. There's like 3 different RNA polymerases, which all do slightly different things. And we haven't even gotten to this next step, which is actually turning the RNA message, sort of intermediary, into a functional protein, which is going to go and do something somewhere in the cell. And that is something that is done by an ancient molecular machine called a ribosome, which is actually made of RNA and protein. So that's like, we'll get into that in a second, but this is maybe a good time to also just talk about the fact that we all evolved from some common ancestor on Earth means that we've been able to figure out a lot about how this process works by studying things like bacteria, E. coli, for example, or yeast. Like we know a lot of our biology from studying yeast. And the amazing thing is when people have gone to try to see if it's the same in humans, it often is the same. Not exactly the same, but the general principles really carry through, because this is something that evolution has tinkered with over billions of years. So at the top here I have genomes and epigenomes, because I'm going to talk a little more about DNA itself. On the left side here we have the actual chemical structures for DNA, deoxyribonucleic acid, or just ribonucleic acid for RNA. And what we can see is A, T, C, and G. So these are nucleotides that essentially, like these are the 2 strands. The 5' to 3' is just the directionality, because everything is complementary. And C's and G's will pair together, and A's and T's will pair together through these interactions, these molecular interactions. They're not actually bonded together, but it's rather like electrostatic forces holding the 2 strands together such that it can be split apart, and you can make templates and copies. You can see that on the right, sort of matching here. Now, what I'm showing here is a chromosome, because you probably are familiar with looking at cells and seeing chromosomes. You have 23 chromosomes. This is essentially just like a higher order assembly, I guess, or like way that the genome gets packaged and organized so the cell can actually divide. Because if a cell is going to divide, it needs to copy the entire genome basically perfectly, or as close to perfect as possible, or else you get mutations and proteins don't work then. So for cell division to happen, we get your genome condensing into chromosomes in a very, very, very regulated process, and then they divide, and that's all done. But this is like a big simplification, because it's not just like DNA is wound up like that, and then just sort of bunches together as a chromosome. This is actually a highly, highly ordered thing, or highly ordered process, which is like really regulated. And when tiny steps in this process are misregulated, say through a mutation that you might have in like a disease, then you can get all kinds of bad things happening in cells. Actually, still on this point, I mean, there's this concept of essentially lethal genes. Like there are, of the, I don't know, let's say 20,000 plus protein coding genes in the human genome, there's a whole bunch that if you have a mutation that means that protein is not made, the cell will just not live. So like an embryo will never develop if it has this mutation, or the cell will die and just not continue. And then, yeah. So I have at the top here from genomes to transcriptomes. So you've probably heard of genomics. It's like studying the human genome. Transcriptomics is the equivalent for RNA, and later on we'll talk about proteomics, which is the equivalent for proteins. So transcriptomics is sort of like RNA sequencing, full-scale identification of all the RNA transcripts inside a cell. What is actually showing here, this is a sort of nice little diagram for the regulation of transcription in eukaryotic cells. So that is cells with organelles, like human cells, or yeast. What this is showing, this is sort of like the actual condensation of chromatin here. And what you're seeing here, there are these round disc-like structures called nucleosomes. And again, this is like an abstraction of an abstraction. And this may be a good time to also say, a lot of what we do in biology is about telling stories about how these molecules are all working together to make life happen. You know, we're going across multiple scales of abstraction, also scale, like size. And some things may seem confusing, but again, just put your hand up if something's not clear. But basically what we have is, in addition to DNA itself, DNA is never just DNA on its own. It is always somehow associated with protein. And DNA plus protein is what we call chromatin, if you've heard the word chromatin before. Those nucleosomes are made of histone proteins. There's actually 8 subunits that come together, and this disk is a sort of just graphical representation of what it is. But DNA winds around this histone with like really specific patterns. Again, we're talking about molecules wrapping around molecules that are all stuck together. And that's what allows it to get packaged up and condensed into chromosomes. These histones also regulate gene regulation a lot. And if gene regulation or gene expression doesn't seem very clear, that's just how all of this works together to express specific genes or not express other genes or turn them on in response to some stimuli or developmental program. I'm not even going to touch developmental biology. I don't know if that's covered anywhere else in the course. But these nucleosomes, these histones, you know, basically the DNA needs to be relaxed for the RNA polymerases to even come in and start reading along, literally physically reading along. Like we're talking about molecules that are going along and then matching the appropriate nucleotides based on the DNA template, which then gets shot out as RNA. There's a whole bunch of other stuff shown on here. I'm not going to talk about promoters or enhancers. These are essentially sequences of DNA, so actual like ATCG patterns of sorts, which we've been able to identify computationally, which tell us where a gene starts, where a gene ends, and which sequences are needed for that gene to be regulated or expressed. What is important here, because I know we're going to be talking about epigenetic stuff later, is histone modifications. So there's all kinds of chemical modifications that happen to these histones, which are proteins themselves, acetylation, methylation. There's definitely others. And that's a whole field on its own. Like epigenetics, epigenomics is not only looking at the DNA sequence, but also these chemical marks that are epi, so like above the genome, but are very, very important for gene expression and regulation and are influenced by all kinds of things. Like, yes, your DNA, your genome is the blueprint for everything, but it's not static. It is changing. Mutations will happen, and depending on what happens to a cell or to a person, there will be marks added to the epigenome, to these histones, to the chromatin, and then DNA itself can also be methylated, which changes its regulation patterns. On the right here, I have a sort of quick run through of all the different types of RNA. So mRNA, or messenger RNA, is probably what you are most familiar with if you haven't studied biology because of mRNA vaccines, because this is where they actually took the mRNA for these viral proteins, put it into the vaccine, and then your cells will take this and create the viral protein, and your cells will identify that as a foreign protein, and you'll develop immunity. Messenger RNA is that exact messenger between DNA and protein, the genotype to phenotype continuum. Other really important RNAs are ribosomal RNAs. So this is actually RNA that is structural. So I'm going to talk about the ribosome, I think, in the next slide. But the ribosome is this ancient molecular machine made of, let's call them 80 different parts, a bunch of different RNA fragments, and then a bunch of proteins. And they all come together in a very, very specific, regulated, ordered way. Like each of those coming together probably has another protein regulating that process. So in this case, RNA can be sort of structural or catalytic even. Another very important one, which you may remember from high school, is tRNA or transfer RNA. So this is RNA that specifically matches up with amino acids. I didn't say this earlier, but proteins are chains of amino acids. But the way that you get those chains and that you match the 3 DNA or RNA nucleotides to a specific amino acid is transfer RNAs. Crazy thing is, a lot of this stuff has only been discovered in the last 50 years. We really have figured out a lot of stuff in cell and molecular biology that really only started to happen after the structure of DNA was elucidated in the mid-50s. Because before then, without knowing the structure of DNA, we couldn't really explain the inheritance of genetic information or what kind of machinery could read this. But figuring out it was a double-stranded helix opened up a lot of possibilities and brought us to where we are today. Finally, if you look up types of RNA on Wikipedia or something, you'll see this massive, massive list, and you'll see so many different things. I'm just going to run through a few others just because I want to help you maybe appreciate how complex this is, because we talk about there being 20,000 protein coding genes roughly in the human genome. But then there's so many other genes which encode for all the different types of RNAs. There are long non-coding RNAs which can sometimes have signaling or regulatory roles or act as structural components in actually the structures that I study inside the nucleus called nuclear bodies, not part of today's lesson. There are small interfering RNAs that are used for silencing and gene regulation. So these are RNAs which will essentially stick to other RNAs and recruit machinery that'll prevent that from being turned into a protein. MicroRNAs as well, small nucleolar RNAs are involved in the assembly of the ribosomes and also of other machinery. It's just they play regulatory roles. And then there's cool things like circular RNAs, which are still being studied now, which we don't really understand, but they're like RNA in a circle. Cool stuff. So the next step after transcription, so you have your mRNA template. The next step is to actually make a protein. And this is what I was talking about a second ago. So this is just a graphical representation of a ribosome, which has 2 subunits. Again, this is made of a bunch of rRNA and a bunch of proteins. And as you can see, there are specific anticodons which match codons. That's the three letters that tell essentially the ribosome which amino acids needs to be put there. And these tRNAs will come in, and this is actually a machine that is moving and doing things and taking this and catalyzing the bond that has to form between 2 amino acids to form this amino acid chain or polypeptide, which will eventually be a protein. There's a lot of terminology here, so you might be thinking peptide versus protein. This can be confusing. I'm going to explain it a little more in the next slide. But this is a dramatic oversimplification of this. I think the Nobel Prize sometime in the mid or late 2000s was for just the structure of the ribosome and explaining how this worked, and all the studies that went from studies in bacteria to yeast to humans to help us understand how this works. This is all stuff that's really only happened since the maybe late 80s and 90s. But we have amazing detail now, and technology is getting better and better every day. Finally, we get to the proteome. And the key idea I want to share here is sequence, structure, and function. So sequence, we can be talking about DNA sequence. We can sequence a genome, and because we know what these patterns that indicate where a gene starts might be, we can start to predict what the different proteins are. So DNA sequencing has enabled us to create this field of proteomics over the last, say, 30-something years. Because that sequence of DNA, we know what protein sequence that will be. And that protein sequence is the polypeptide chain of amino acids. And that's what we call the primary structure. So this gets made by a ribosome. Then we have these concepts of secondary structures. Just the molecular interactions between these amino acids will sort of force them to fall into these general shapes, like alpha helices and beta pleated sheets. Again, very, very simplified example here. But this has to do with thermodynamics. Like these molecules are at a lower energy state here, so they naturally want to form these structures. Tertiary structure would be not only these secondary structures, but the whole thing all folding together. And then quaternary structure would be multiple subunits coming together. A lot of proteins exist as complexes. They're not just one protein alone. They are 2 or 3 or more proteins all fitting together and making a much bigger machine. Like I talked about the ribosome, I think there's 80 plus proteins in there. Just to do this single, very, very, very important step in the expression of genes. Yeah, so I mean, you've probably heard of AlphaFold, AlphaFold 2. These are amazing, amazing pieces of technology that honestly solved a problem that I didn't think would be solved in our lifetimes based on just the sheer computational complexity of predicting how a protein is going to fold. So we're talking about complex molecular dynamics that are hard to predict. And also, they don't happen just in like a constant stepwise way. And like there's whole courses on just protein folding. Like this, I'm like barely scratching the surface here. But yeah, essentially, the actual structure of the protein determines its function. Like how it folds up, the 3D shape it takes is going to determine what it is doing. And that's just a very important thing to understand. Like I said before, your proteome has about 20,000 protein coding genes. But there's this thing called alternative splicing where mRNA gets made, and then there's other proteins which come in and splice out, just like, you know, if you imagine splicing a reel of film or something, take out different sections, and this then changes the pattern, right? So there will be sections of the protein that are missing, and this can form a completely different protein, or maybe a slightly different protein, which just with this like, you know, small change now goes to one organelle versus the other. And then finally, when we talk about PTMs, so this is a term that I think will probably come up later on, post-translational modifications. These are chemical modifications that happen to proteins post-translation. And there's like countless. We haven't even discovered all of the different PTMs. Like there's a few very important ones like phosphorylation that you might hear about, or ubiquitination, but there's just like so many. Glycosylation, so carbohydrates being added on to these proteins, like I said earlier, that's one of them, and that's extremely hard to detect and measure. Like everything in biology at this scale, we're talking about molecules, and you can't really measure things directly. Like whatever you're doing, you're sort of inferring it using other biochemical techniques and trying to make sense of the system and setting up all these careful experiments, because you can't just look at the cell and see what the molecules are doing. So when you take into account all the various PTMs, there's probably millions of different unique proteins inside your body. And we know what, you know, a couple % of them do maybe, or have an idea based on homology to other organisms, again, evolution. But this is just like, yeah, it's crazy. We're talking about, in the case of longevity, wanting to fix something that is broken, but we don't even know what most of the parts are doing. Like we know they're there because we can read the genome and we can predict that this will create a protein, and we can even try to express those proteins and see if they fold. But how it fits into the, you know, complex machinery of a living organism, we have no idea. And this is why I love biology, because it's beautiful and it's fascinating. And it's, I saw a line on the last slide before I went on, but it's like staring at the night sky. Like we can see all these things there and we don't understand them exactly, but we know they're there and we know they're important because over billions of years, evolution has shown us that these things are important. So yeah. This is a very complicated diagram, which is really low res on here. And what this is describing is the actual assembly of ribosomes. So this is like a very, very core basic thing that all cells need to do constantly. They're constantly making ribosomes, constantly making protein, constantly making mRNA. Like these things are shown as stepwise reactions, but they just happen as a continuum. It's a treadmill. It's always going on. It's happening in you right now. I told you that ribosomes are made of ribosomal RNA and proteins. Essentially, they start their assembly in the nucleolus. If you guys have heard of, if you remember the nucleolus, the nucleus in the nucleus, that's actually the site of ribosome biogenesis. And it's also where our RNA specifically gets transcribed by, I think, RNA pol 3. So there's a few different rRNAs. They have their own processing, as you can see here. So this is the nucleolus. This is the cluster of our DNA genes, I guess. These are the different pre-ribosomal RNA pieces that get made. 18S, that refers to the size when you're doing centrifugation and trying to get some gradients. Then there's all sorts of modifications, which are helped by things like SNO RNAs, again, other RNAs, working on these RNAs to help them get to their final processing point. These then get sort of like shuttled out of the nucleus into the nucleoplasm, so that's like the stuff inside the nucleoplasm, which we contrast with the cytoplasm, which is everything else inside the cell. They link up with various proteins, which make up the ribosome, and then these two subunits get exported to the cytoplasm so they can form an actual, Siri is acting up, form an actual working ribosome which can make proteins. And just like, I'm not going to talk about most of what's on here, but there's all kinds of interesting things to point out, just again, complexity of biology. We are talking about these very complex machines being assembled in a really highly tightly regulated process, but the proteins that are made for this are made by ribosomes in the cytoplasm. So because translation is happening in the cytoplasm, all of the proteins for ribosomes have to be imported through another separate, very regulated process. And this is like a whole field on its own. So that way you can even form ribosomes. Like there are people who spend their entire lives just studying how pieces of ribosome assembly work. And it's cool, because it's like a molecular machine that allows life to happen. But yeah, this is just like a sneak peek of how this all kind of fits together and why spatial separation is important, but also temporal separation. I remembered another point I wanted to make on the structure and function side of things. As proteins, like we again, like to tell these stories in very stepwise ways, but it's all happening at the same time. Like it's not like a polypeptide is made and then it folds with the help of other proteins called chaperones. But this is like, as soon as the polypeptide chain starts coming out, other chaperone proteins are interacting with it. When I say interacting, I mean physically like bumping into it and forcing it to start folding and like testing out what's the proper conformation. So this is, I think, the last slide I have, and it's more of just like a, I don't know how much time we have for questions or anything. No time, okay. Just sort of like a high-level overview, because I was supposed to talk about biochemistry in this lecture, but biochemistry is essentially chemistry happening in living things or of living things. And I just wanted to run through a few different of these like fields in biology to maybe help you understand what the distinctions are, or just like how much overlap there is. Like my PhD is in the Department of Molecular Genetics, which is like saying biology. It's so many different things. You know, we talk about biochemistry, and often that would be people maybe studying how enzymes do things, but it could also be just like reactions on different, I don't know, metabolic pathways, which you'll be hearing about from the next speaker. But then we can talk about biological chemistry, which is more like people doing chemistry, like more hardcore chemistry, just on biomolecules. And then chemical biology is like chemical screens on living systems, so it's like a separate thing altogether. Molecular biology often refers to what we were just discussing, biology at the molecular scale, so different pathways, how these things fit together to do things, mechanisms, usually like multi-molecular, multi-party systems. Cell biology is just like the scale that we're looking at this, and often people will be looking at, I don't know, how proteins move through the cell, which organelles they go to. But then we have so much other stuff. We have like genetics, genomics, proteomics, computational biology and bioinformatics, developmental biology, immunology, and these are just like different focused systems that, again, rely on all of the same principles. The nice thing about biology is it all has a common ground. So the more about it that you learn, the easier it becomes to learn more stuff. Like once you have this foundation, you can learn about immunology or developmental biology really easily, because all cells have proteins and DNA and RNA, and they all have to do translation, and they all have to do signaling. So proteins doing things to other proteins that, you know, put PTMs on them, which change the conformation and turn them on or off, acting as switches. So yeah, I think that's it. That's my really fast overview of life at the cell and molecular level.
So hi everyone, I'll be taking the next couple of sessions. So before I just get going with my topics, I thought I should just introduce myself. So it says Sheekey on the board, that's my surname. My name's actually Eleanor. So there's kind of like maybe a few things you need to know about me. Firstly, I love biology, that's my favorite subject. Secondly, I'm currently a PhD student at the University of Cambridge, so in England. I've been studying biology for now almost probably like 10 plus years. So the fact that you guys are going to go through a lot of topics today is very impressive. I'll try and keep things as simple as possible, but like Boris was saying, if anything doesn't make sense, I just love talking about biology, so come find me afterwards and I'll explain it, hopefully in more detail. And then I also have a YouTube channel, so some of the concepts we go through today, I have videos that might illustrate it a bit better, like they have more animations if you prefer that sort of way of learning. So if you want to learn about that, also come find me and I'll happily talk for ages about it. So somehow I've been given the topics of metabolism and homeostasis, which are both actually quite big topics. So let's see how it goes. So metabolism, where to begin? Now, as I said, I'm from the UK, and in the UK, we're somewhat proud of our underground system. And so this is a picture of the London Underground. You might be thinking, why am I showing this? Well, in many ways, the best way to think about metabolism is just a series of different interconnected pathways. And so like the London Underground, you want to know how to go from A to B, and you can find some sort of different pathways in which you can go from these different molecules. So we learned about DNA, we learned about proteins, and we learned about lipids and things like glucose and other substances that we intake. And basically metabolism is just the big overview of how we go about modifying these different chemical components and either build things or break things down. And so Nathan actually said at the start that there's going to be a test, so I hope you're paying attention. Now, this is me trying to be funny, but that is a somewhat overview of many different metabolic pathways that you can find within a cell. Now, as I said, I've been doing biology for 10 plus years, and I don't know fully that circuit map. So don't worry, you don't really have to understand it. But what I want to emphasize is that metabolism is complicated, and we are still trying to understand how the cell builds things and breaks things down, and also understand the different enzymes, so proteins, that are actually catalyzing these different reactions. But for this session, all you really need to appreciate is that there are these different pathways, and that by and large you can split them into two categories. Either the cell is making things, so anabolic pathways, or the cell is breaking things down, catabolic pathways. And yeah, so issues with this diagram is that it's too complicated for this session, in fairness. It's probably wrong anyway. We often make new discoveries and realize that there might be a new pathway that we've not yet discovered, or that maybe we have a pathway but we're missing steps. And the other thing to really understand is that metabolism is dynamic. It's happening all the time. Some pathways are going to be having a lot more activity than others, and it's just highly regulated. And so to go back to my London Underground example, this is just a snapshot of like the density of people using the tube station and how some pathways are more active than others, and it can vary during different times of the day. So this is currently showing the activity of different tube lines in the early hours of the afternoon, but it looks quite different when it comes to the rush hour. So the idea is that metabolism is complicated, it's dynamic, but essentially it's just a series of different pathways that are either building things or breaking things down and just modifying chemicals. So this is just a snapshot of like one cell in time. That's probably incomplete. So we talked about these pathways, but what exactly is it that cells are making? So the anabolic building pathways. So essentially, you can split that down to some pretty key components for a cell, and that firstly is DNA. DNA being made of a series of different chemical nucleotides, so these A, C, T, and Gs. And so they need to be made within a cell. The cell also makes proteins. These are made of amino acids, as we've just mentioned, and so they need to be built within a cell. And also the lipids. There's a massive variety of different types of lipids that make up various membranes within the cell, and they also need to be made. So the cell's busy making a lot of things. So how do you go about making things? Well, the cell needs some energy to do this. And so energy, in terms of biology language, you may have heard of this component called ATP. Now, this is the structure, well, a very simplified structure. And essentially, ATP stands for adenosine triphosphate. Don't really need to understand it, but the triphosphate refers to the fact that there are these 3 phosphate groups added to it. And to release energy, ATP is broken down to ADP, so diphosphate. So this release of one phosphate molecule enables the release of energy, which facilitates a lot of different reactions within the cell, in particular these metabolic pathways. So we've now gone through, probably quite briefly, different pathways in terms of building substances within a cell, and that requires energy. But where does that energy come from? Where do we get this ATP? And that actually comes from different catabolic processes, so the way that the cell breaks things down. And so the classic sort of pathway you're taught about in biology is that we take in glucose, and that glucose is broken down to release energy, and that energy we capture in the form of ATP. So this is a very, again, simplified, but for this introduction, this is all you really need to know to understand the basics of biology, is that we intake glucose, it's taken up into our cells, it goes through a series of different pathways where it's rearranged into different chemicals, but during this process, energy can be released in the form of ATP, and it's a very efficient process. And so essentially we take in glucose, it goes through this series of pathways, we get ATP, and then that ATP can be used for all sorts of things within a cell. So enzymes, so proteins use it to perform anabolic pathways, but also just for the activity of proteins themselves. And as I haven't mentioned it, this is the mitochondria. So often we call the mitochondria the powerhouse of the cell, and that's mainly because that's where the majority of this ATP is produced within a cell. So we spoke about making things, we spoke about breaking things, and often these pathways, they can happen at the same time. But it'd be very inefficient if they were happening both at the same time, as you'd end up with these sort of futile cycles where you could be breaking something down and then making it and then breaking it. And you might think that's quite wasteful. And so essentially, as well as understanding that there are these different pathways, the pathways themselves also have to be tightly regulated. And so to go on from the example of glucose, I wanted to explain how glucose metabolism is also tightly regulated and requires the use of different organs within the body. So in a high-level, sort of simplified version, the liver is an organ where the cells can essentially not just break down glucose, but they can also remake glucose. And they can deliver it into the bloodstream so that other organs, like the muscle, can use this glucose and break it down to get energy in the form of ATP. And so these different organ systems and these different organs are communicating with each other, and there's many different levels of control within such a simple system, which is just the breaking down and making of glucose. And so this is when you have to also appreciate that we sort of subdivide these different divisions of biology into different sections, but actually there's a lot of overlap between them. So metabolism itself is also linked with this idea of homeostasis, which is the idea that a cell tries to remain within a somewhat stable state. So our cells are always fluctuating, responding to external stimuli as well as internal responses. And essentially, the cell wants to stay in this sort of healthy, in quotation marks, state. And if it goes too far beyond that state, we can consider the cell to become more diseased. And so it's how does that cell maintain within these boundaries and stay healthy? And it sort of oscillates between the two. And to do this, you have to have ways of detecting these changes, so different receptors. There has to be some element of interpretation of that change within the cell. And then there has to be some effector to have a response to then bring it back to that baseline level. So for example, in the case of glucose, or firstly, in the case of temperature, we all know that when we get too cold or too hot, we suddenly have all these different responses that happen. And essentially, that's all trying to get us back to the same set point. So yeah, I don't think I need to explain that when we get too cold, we start to shiver. If we get too hot, we start to sweat. And so that's just a couple of examples of ways in which there are responses built into our system that help to bring us back to this level of homeostasis. And that if there's issues in getting to homeostasis, that can often lead to diseases. So to come back to glucose regulation, as we eat a meal, we will notice that our glucose levels will spike. And how does the cell respond to that and bring it back down to ideal levels is something that I'm just going to briefly run through as one example to illustrate this concept of homeostasis. So essentially, what we need to appreciate is that glucose regulation itself is teamwork. So far, we've only really thought about things at the cellular level, but we now need to sort of take a step back and realize that this is a whole system that we're working with, and that it actually involves communication between lots of different parts of the body. And so I'm not going to go through this whole slide because it's just too much information for this introduction. So I'll give you a more simplified overview. But to do that, I just want to reiterate this point about the different levels in which we talk about in biology. So far, we've only really spoken about cells. And so cells sort of, they can work together essentially to build tissues. And so tissues, they're not like things people are familiar with. Like, there are different types of tissues within the body, and they all have different sort of characteristics. And that obviously depends on the cells that make up the tissue. But then you can have different tissues work together to make organs, and organs work together to make organ systems, and then organ systems work together to make humans or any individual. So the idea is it's all about teamwork and communication between these different components, which are actually really important. And that sort of also goes to show like how hard it is to then study these processes, because a lot of biologists, including myself, we work at the cell level, and it's very easy to get lost within that cell level and forget how to take that step back and appreciate that. How does that fit into the bigger picture about how it's working together with other cell types, and how it fits into understanding its role within tissues and organs? And when you then go to say, well, how do humans survive? How do we live? Why are we aging? These sort of questions, it's like, where do you start? And so this is just a sort of one way in which we can start to break this down, is to appreciate that there is a sort of hierarchy in how we're built. So maybe I will go back to the slide to mention glucose regulation. So essentially, the key sort of players is firstly the liver. And so if we've just taken in a meal of glucose, the liver can essentially use some of that to produce its own ATP, but it also stores it in any excess as like glycogen within its cells. And so that's useful because when you're now going for a run, your muscle, it needs glucose to break down to get energy. And so what happens is that while the muscles are breaking down glucose, the liver can then convert its storage back into glucose and then put it into the bloodstream so that the muscle cells can then take it up. And so that's just one very brief example about how there is this massive communication between different organs. And that even something as simple that we already pretty well understand, such as glucose regulation, is already quite complicated, but fascinating. Which leads me on to another very complicated thing that I've been tasked with explaining, which is the immune system. So again, the immune system is something that involves many different parts of the body. For the purpose of this, I don't think you really need to understand much more than the fact that we have this lymphatic system, which is one way in which the immune cells can sort of transport. So this is just to illustrate that the immune system is complicated, essentially. But I'm going to break it down into how I try to understand the immune system or try to explain it, which is firstly to appreciate that there are these two sort of main categories, innate and adaptive. So I think we all know what it's like to be ill and to overcome a cold. And so initially we have sort of fast and very classically non-specific pathways, which we refer to as the innate immune response. And one often unappreciated feature includes just having the skin as like a barrier to a lot of different pathogens that otherwise could invade our body. And so the immune system is just a very well mechanism that's effectively trying to protect our body from different pathogens, bacteria and viruses, but also from cells in our own body that might become tumorigenic or cancerous. And so the innate system is comprised of not just like the skin and like physical barriers, but also actual immune cells, which I've illustrated in this diagram here, which essentially just sort of try to sense these sort of foreign particles within the body and kind of trigger to the body that something's here that shouldn't be here, do something about it. And so while that's very quick and quite non-specific, effectively we also need to employ the adaptive immune response to mediate a more effective reaction to whatever's infected or invaded the body. And so this is when we come across things like the T cells and B cells that you might have heard about, and the production of antibodies. And so antibodies are very interesting proteins because they're very specific to certain antigens. And so antigens are things that are found on foreign particles. And essentially, the antibodies are very useful for providing a variety of specificity to different types of invaders. And once we have the antibody release, that helps to boost the immune response and activate the T cells. And T cells themselves are complicated, so I'm not really going to go too much into it. But essentially, some of them can also kill the infected cell. Some of them are used for memory, so if you get invaded again, they're ready for the response. But the fact is that this is a prolonged series of pathways. It is a slower process, which is why it often can take a while for you to overcome a cold. But it's more specific and tailored to whatever cells are infected, and therefore it's going to be targeted to those damaged cells as opposed to it sort of also harming healthy neighboring cells. So this also ties into homeostasis because we don't want to be constantly stimulating our immune system. We want there to be a sort of acute response. So that is, we know that there's something wrong, that the immune system gets activated, it deals with the problem, and you go back to the sort of resting state. Now, when we come on to talk more about aging later, sometimes you'll hear the words like chronic inflammation. And so that's when the immune system gets stimulated and it sort of remains stimulated and continuously inflamed. And so therefore, it's kind of a loss of homeostasis in this situation. And so speaking of chronic inflammation, sometimes you often read that we want to boost our immune system, we want to make it stronger. But actually sometimes maybe we don't want to do that because having a strong inflammation is already linked to things such as autoimmune diseases, where there is this response to healthy cells, and that can lead to things such as type 1 diabetes. And also a lot of diseases of aging, which we'll probably come to talk more about later, are also associated with inflammation. And so this slide just sort of only highlights a couple of diseases where inflammation is strongly linked with the disease and also thought to catalyze and sort of progress the disease even further. So as we'll sort of appreciate later, all of these sort of conditions where this is chronic inflammation are tightly linked with aging itself. Now, besides getting me to explain metabolism and the immune system, I've also been tasked with telling you what we don't know. But that's actually not too bad because, to be fair, there's a lot of things we still don't know in biology, which is great for me because that gives me a job. And, well, also means that there's still a lot that, we have to understand. And so this diagram is just showing that we still don't fully understand what life is. Like, how do we go about defining life? How did life originate? If we're going to be trying to address questions like why do we die, why do we age, some of that could be reflected in at least appreciating what enables life to survive. There's also developmental biology that Boris briefly mentioned, which is how do we go from a single cell to a fully grown organism? That's obviously been somewhat mapped out, but something that we sort of don't really understand. The reason why it is now interesting to look back at this very early stage of development is of particular interest for things like regenerative medicine, like how could we go about replacing different limbs or different structures that happen in certain organisms like salamanders, but could be really useful in terms of therapeutic application. And we still don't know how that happens, and I think it's fascinating. So that's another thing we don't know. we also don't fully understand how information is read within the cell. Now, often people think that, oh yeah, we have the human genome sequence, surely that's what all the information is, but that's not quite true. We also, we don't understand how the cell reads that information. We're trying to understand the language of life ourselves, but having just the genome sequence doesn't tell us everything. In fact, when the human genome sequence was fully published, we all thought that that was going to be the savior for biology, but here we are 20 years later with now actually a fully sequenced human genome, and we still don't know the answers to these questions. So clearly there is information elsewhere, and that's reflected in the other components of the cells, such as RNA, but also things like metabolites that are found, and ions, and how things are communicating with each other. And so essentially we don't have a full understanding of information and communication systems within a cell. And this leads me on to some work done by one very interesting researcher, Michael Levin, who's essentially kind of asking these really big questions, but is actually making some headway with some of the research. In particular, he's interested in regeneration and understanding like how can we go about building organisms, but without having to really understand the intricate details, but just having an understanding of what the high-level triggers are. That means you could intervene higher up and just sort of let the sort of systems already in place do what they do best and not have to worry about the intricate details. And so he's sort of pushing for this concept of bioelectricity, which is how a lot of, well, all of our cells have a so-called membrane potential. So there's different positive and negative charges within the cells, and that's also used as a mode of communication, but also a way to pass on information within a cell. And so it's a whole new interesting area being explored. And so a lot of this goes back to what we were saying earlier about the different levels of biology, that now to ask these sort of questions, we really need to take that step back and understand the bigger picture and go find model systems and like actual methods to understand experimentally how these different tissues and organs communicate together. So as a biochemist, what we usually would do is basically break open a cell and see what's inside the cell. But that's great, but that's just one snapshot, and these systems are dynamic. And so we need to build better tools as well to sort of understand what's really happening. And then this is a picture of the brain. There's many things we don't know about the brain. How is memory stored? How do we think? What is consciousness? How do things like Alzheimer's disease progress? It's something, yeah, just too many things we don't know about the brain. And then as to sort of lead on to the topics that we'll have later today and the next few days, we still don't really know what's driving aging or what the main causes are. And so this is also kind of one of the interesting things we want to find out. And hopefully this will lead us on to some of the talks we have later today. So yeah, I realized I've gone through a lot of topics, probably quite fast. But if you want to have a better understanding or some better drawings, come find me, because I would happily talk about it. But hopefully this has given you at least an appreciation for the different systems that are going on within a cell. And that is complicated, but if you have just a few basic principles under your belt in terms of understanding biology, you can then sort of do your own reading and go a bit deeper. But hopefully it's also excited you about some of the things we're working on in biology, and that it's really quite interesting. So I do think that's the end of my slides.